Hi everyone, so today we're going to cover microbial genetics and metabolic regulation as well as some eukaryotic facts to kind of help you distinguish the difference between genetic events in prokaryotes such as bacteria versus eukaryotes such as us. So the first thing we have here is just a reminder of the nucleic acids. And you'll notice that there is this little area here with the pyrimidine and the purine bases that are part of nucleic acids. And the trick to remembering which is cut pie. So like you would cut a pie, think C, U, and T are all pyrimidines, whereas A and G are your purine bases. And the difference between them you can see is pyrimidines are one ring, whereas purines are two ring structures. Okay? And it's kind of funny because the bigger word is the smaller structure. Okay? And then we also have here for you to see the phosphate sugar alternating backbone of DNA and RNA. And then here the full structure where you can see the phosphate, sugar, and nitrogen bases. Now on this slide what we're going to focus on is the gyrase or topoisomerase, which is an enzyme that you find in prokaryotes but not eukaryotes. And that's very important because since gyrase is involved in coiling and uncoiling DNA in bacteria, which is very important for their DNA replication, but it's not found in our human cells, in microbiology, that makes gyrase a very good target. You're able to kill bacteria if you inhibit gyrase. And you don't hurt the human because, well, we don't have gyrase. Okay, so make sure you know what gyrase does, which is coiling and uncoiling DNA. But also make sure you know that it is a good target to kill bacteria. Now, when we talk about genetics, one of the important things to always remember is what is the central dogma of biology. So anytime you see the term central dogma, I want you to write down DNA to RNA to protein, meaning that the way that genetics flows in biology is DNA is transcribed to RNA, which is then translated to protein. Now what I have here is just a little figure for you to see the processes that we're going to review today. The first one over here shows you DNA replication, and then you have that DNA being transcribed into RNA over here, and then you have that RNA getting translated into protein here. So this figure here nicely demonstrates the central dogma of biology, going from DNA to RNA to protein. Now, before we get into the details of each of the processes, I just want to show you a couple of videos to help you visualize and remind you what's going on in each of these processes. Because at this point, you should have encountered them in other classes as well, so we won't spend too much time going into the details of each one. So first up, we have a general overview of DNA replication.
In order to pass genetic information on to its offspring, an organism must make a copy of its DNA. The process of copying DNA is called DNA replication. During replication, each strand of the parental DNA serves as a template in the creation of new DNA. Because each newly synthesized DNA molecule is made up of one parental strand and one new strand, DNA replication is described as semi-conservative. One strand in each molecule is conserved, while the other strand is newly replicated. In many organisms, the two DNA polymerases responsible for replicating the leading and lagging strands are linked together by connector proteins. To make this possible, cells take advantage of the flexibility of DNA. The leading strand is synthesized continuously toward the replication fork in the 5' to 3' direction. Since the leading strand is synthesized continuously, it only needs one RNA primer to start the process. Since DNA polymerase can only add nucleotides to the 3' end of the growing DNA molecule, the lagging strand is synthesized discontinuously away from the replication fork in the 5' to 3' direction. The DNA of the lagging strand is synthesized in pieces called Okazaki fragments, which consist of about 1,000 nucleotides. Each Okazaki fragment requires its own RNA primer.
So as that video reminded you, when we talk about DNA replication, you're producing two DNA strands that are identical copies from an original double-stranded DNA. Now the term you have to remember for this process is semi-conservative replication. So ask yourself, what does it mean if it's semi-conservative replication? Well, it means that the original DNA is partially conserved, because in DNA replication, both strands will now serve as templates for the new DNA strand. So each of the daughter DNA molecules will have one strand that's the old original DNA, and a second strand that is then the newly built molecule. So that's why it's called semi-conservative. The other thing that you always want to ask yourself is, when does a cell need to replicate DNA? And we touched upon this in the previous lecture as well. A cell needs to replicate its DNA before mitosis. Okay, so always write that down, put stars, circle that. A cell needs to replicate DNA before mitosis, because if you think about it, if a cell is going to split, if it doesn't double its DNA first, then each of those new cells is only going to have half of the genes that they're supposed to. Okay, so it's very important that cells double their DNA first, before mitosis. So now to recap the specifics of what actually occurs in DNA replication, and for any of the genetic processes, if you understand what each of the enzymes does, then you would be able to explain the process to someone. So with bacterial DNA replication, the first two things you need are gyrase and helicase. We just mentioned a few minutes ago that gyrase is responsible for uncoiling the DNA that's currently all coiled up tightly in order to fit inside that little bacterial cell. Once gyrase uncoils the DNA, then helicase unzips it, meaning it denatures those double strands into their single strands, so that each strand will now be able to serve as a template. Now, when you have those double strands separated into single strands, any single nucleotide or nucleic acid basically is very single. and clingy and very much wants to find its partner. So what happens is, if you don't do something to prevent it, those single strands would stick right back together. So that's why the cell needs single strand binding protein, or SSB. Single strand binding protein you see in this diagram here. Once the two single strands are opened up, SSB coats each one to prevent the single strands from sticking right back together. Okay? Now we're going to move to the next slides to then explain the final enzymes that you see listed here. Once the strands are separated, now you can have polymerases come along. And anytime you hear the term polymerase, I want you to think an enzyme that builds the nucleic acids. So a DNA polymerase will build DNA strands, whereas an RNA polymerase will build RNA strands. Now, DNA polymerases have their limitations. They're not that great at doing their job of building DNA. Their limitations are that they are one-directional and they can't start new strands. And when we say one-directional, that means that a DNA polymerase can only build a DNA strand from 5' to 3'. Okay, and remember, that's simply how we label DNA orientation. 5' to 3' is the only direction it could go. So that makes us wonder what's necessary to compensate for these limitations. If DNA polymerase can't work in any other direction and can't start new strands from scratch, how does new DNA get made? Well, that's where primase comes into play. Primase is a special RNA polymerase that will make little RNA primers, which you see in the figures here. Little RNA primers that will serve as a starting place for the DNA polymerase to then build off of. Okay, because RNA polymerases do not have limitations. They can start a new strand completely from scratch. They don't need any other base to build off of. So when you think of the order of enzymes in the process of DNA replication, we had gyrase and helicase, then we had SSB. Now we have primase comes along and puts the RNA primers, and then DNA polymerase builds the new DNA strand off of that little starting primer. Now that then leaves you with a problem at the end of DNA replication. Well, now you still have these little pieces of RNA in the double-stranded DNA. That's a problem because, as you already know, RNA has some different base composition than DNA. So RNA will have uracils in it, and it will lack any thymines, which would be a problem. So at the end of DNA replication, you need pol 1, RNase H, and ligase. What happens is pol 1 and RNase H, they degrade the RNA primers, meaning they get rid of all of the RNA bases. Pol 1, which is a polymerase, then replaces those bases with DNA bases. The last thing to happen is ligase, which serves as a little glue, will fill in any gaps in the backbone. So the ligase will basically glue in the new DNA bases. Okay, so again, what happened was the RNA gets removed, it then gets replaced with DNA bases, and ligase then does the final gluing of the backbone to fill in the gaps. Okay, so let's recap that one more time. The way DNA replication works is, first you have the gyrase, and the gyrase uncoils that big ball of DNA. Then the helicase comes along, and that unzips the double strands so that there are now two exposed single strands. Then SSB comes along and coats both of those single strands so that they don't instantly stick back together. Now that those strands are nice and separated, primase can come along, and that's the one that we said is a rock star. It's an RNA polymerase that will start off our new strands, but it's starting off as RNA bases, just a few, usually about 6 to 9 bases. And now that those bases are there, DNA polymerase, which has its limitations, now has something to build off of as a starting point. So now DNA polymerase can take over and build the rest of the DNA strands, putting together those A's, T's, G's, and C's. Once that's complete and we have our 2 new DNA strands, there's currently a problem, right? After DNA synthesis, there's that problem that there are still some RNA bases in that DNA strand because the primers are there. So now PO1 and RNase H will degrade or remove any of those RNA bases, and PO1, which is a polymerase, will now build or replace those RNA bases with DNA bases. The last thing to happen is ligase basically cleans up the last minute messes and will reseal any little gaps that are currently in the backbone of the DNA. And boom, there you have the bacteria has 2 new DNA strands, which are semi-conservative. They are made up of 1 original DNA strand and 1 newly built DNA strand. And all of this occurred before mitosis, okay? Before the cell needs to divide. So that after mitosis, each cell will have the proper amount of DNA, okay? If you have any trouble with any of the enzymes or any of the DNA replication process, just send me a remind message and I'll give you a bit more information. Again, this is meant as a chapter in microbiology that is supposed to be review, but I understand that some students may not have had this before. So feel free to ask as many questions as you like. You are never bothering me. So now we've reviewed DNA replication, and now the cell has divided. It's nice and happy with the right amount of DNA. Now it's time to actually use that DNA. And whenever you see the word express in genetics, that simply means to transcribe. So we're going to go through transcription now so that the DNA that's been replicated can now serve as a template for RNA.
Cells use DNA to store information, while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules, which can be used to synthesize specific proteins. Cells use 3 types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes. There are three stages in the process of transcription: initiation, elongation, and termination. Let's see how each stage works. During initiation, RNA polymerase binds to the DNA and recognizes a site called a promoter at the 3' end of the template strand of the target gene. When RNA polymerase finds a promoter, it breaks the hydrogen bonds holding the DNA strands together at the site of the promoter, and transcription begins. RNA polymerase does not bind to all promoters with equal affinity. This difference in promoter strength is one way that cells can control gene expression. The more strongly RNA polymerase binds to a particular promoter, the more likely that gene is to be transcribed. In general, higher levels of transcription lead to higher levels of translation, which leads to higher concentrations of that polypeptide. One strand of the DNA serves as a template strand. The RNA transcript is copied from this strand of the gene and therefore has an RNA sequence complementary to this strand. During elongation, RNA polymerase moves along the DNA, opening up a bubble in the DNA. As it moves, RNA polymerase adds bases to the 3' end of the growing RNA transcript. Elongation continues until RNA polymerase reaches a site on the DNA called a terminator. At this site, RNA polymerase and the newly synthesized RNA transcript are released from the DNA. Termination can occur through two mechanisms: self-termination and enzyme-dependent termination. In self-termination, the RNA sequence transcribed at the terminator causes the RNA to hydrogen bond with itself, forming a stem loop structure, which essentially pulls the RNA polymerase off the DNA. In enzyme-dependent termination, a termination protein binds to the terminator and pushes RNA polymerase off the DNA.
So as you saw from the video, with transcription, just like you'll see with translation, you have the three terms: initiation, elongation, and termination, and you have to know what's going on in each of these steps. So initiation is how things get started. So with transcription, there's two key players in prokaryotic initiation. The first one is the promoter, and next to the word promoter, I want you to write DNA sequence. So the promoter is a DNA sequence that basically tells the RNA polymerase, hey, I'm a gene that needs to be expressed, come here and bind to me. So the promoter is what gets recognized by RNA polymerase in order to be expressed. Every single gene needs a promoter in order to be expressed. Otherwise, RNA polymerase, which builds the RNA, would never be able to arrive at that gene to transcribe it. Now, the RNA polymerase that's responsible for actually transcribing or making the RNA is made up of two subunits, and there's a trick to remember which subunit's doing what. For sigma, I always write sigma sees the promoter. So I underline that S and think sigma sees the promoter, because that's the subunit that actually recognizes and binds to the promoter to start transcription. Whereas the core, that's more involved with elongation. The core constructs, okay, that's how I remember it, core constructs in order to build the rest of the RNA. Okay, so sigma sees the promoter and then the core constructs, and the core constructing is more the elongation step. So just to make sure that you have the memory trick written down in your notes, I've written it on this extra slide here. So sigma sees the promoter, and so it starts transcription. That's part of initiation. So whenever you're asked about initiation of transcription, remember, sigma subunit of the RNA polymerase and the promoter. Okay, those are the two things involved in initiation of transcription, whereas elongation, core constructs the new RNA strand, and that is elongation. Okay, so this is just another recap of initiation and elongation with a couple of extra details. So basically what happens is sigma sees the promoter of the DNA, it binds, and the core enzyme will then open up the DNA. 6 to 9 bases will be synthesized while at the promoter, and then at that point sigma is no longer needed, and so it pops off, whereas the core continues elongating the new RNA strand. Okay? And the reason why I write RNA pol is the rock star is to once again emphasize that RNA polymerase does not have the limitations that we saw with DNA polymerase. Now, with everything in life, everything that has a start needs to have an end. So for transcription, termination occurs when the RNA polymerase hits what's called a hairpin structure. And this figure right here shows what a hairpin loop looks like. And basically what happens is the RNA polymerase gets destabilized by that hairpin loop, and it causes it to pop off. Okay, so when you think about transcription, remember initiation is the sigma subunit and the promoter. Elongation is the core subunit of RNA polymerase. And then termination is the hairpin structure that destabilizes RNA polymerase. Now, when you go through all of this, students a lot of times just think of RNA as mRNA because that's what they know codes for the protein. But always remember that tRNA and rRNA are just as important. They all have a role in translation, which is where we're now going to make the protein. Now, to remind you with translation, again, you're going from nucleic acid to protein. And in this process, mRNA is the code that will basically tell the ribosome what amino acid needs to be where. rRNA is very important. That's actually part of the ribosome. And the fact that the ribosome has some rRNA, which is nice, clingy single strands of RNA, that allows the ribosome to grab a hold of the mRNA to then use it as the template. You then have tRNA, which is going to be what's actually holding the amino acids and brings them together so that the ribosome can build the protein. Now with tRNA, sometimes you'll hear it referred to as charged or uncharged. Charged simply means that the tRNA currently has an amino acid, whereas uncharged means that the tRNA gave away its amino acid, so it's no longer holding any amino acid. Now, one other thing I want to remind you of on this slide is the fact that this ribosome structure over here, which, like I said, that has some rRNA on it. So this ribosome structure is what we call a nucleoprotein. Nucleoprotein is important because, again, the fact that the ribosome is protein because it's an enzyme, but also has some nucleic acid, the rRNA, allows it to grab to grab mRNA, which is important. Now, the ribosome has 3 sites on it. It has A, P, and E site, so you could remember it as APE or EPA. The A site is the amino acid acceptor site, so the tRNA that's in an A site will have just a single amino acid. It'll be charged. The P site is the peptidyl or peptide site, so the tRNA that's sitting in the P site will have the growing polypeptide string, the growing protein, and the E site is the exit site. So that's where the uncharged tRNA will then leave the ribosome. This will become much clearer in a minute when you review the video. Okay, this slide is just so that you can pause the video and write down the notes in case you didn't catch what each of the sites were. So now just to go over the 3 main steps of translation before you visualize it in the video. Just like transcription, there has to be an initiation, an elongation step, and a termination step. So with initiation, what we have is the ribosome being in charge of the grabbing hold of the mRNA. And the way it does that is mRNA will have what we call a Schindel-Garno sequence on it. And that complements the piece of our RNA that's part of the ribosome. So like I said, complements, single strands, they're very sticky and want to match with each other. So this part here, this is the ribosome, and it grabs a hold of the Schindel-Garno of mRNA. That's part of initiation. The other part of initiation is the fact that the mRNA has a start site that matches the tRNA that will bring over what we call the initiator tRNA, which is fMet. Okay, fMet will always be the first amino acid in any protein built. Now what happens is, before I put the bullet points out for you to see, I kind of want to help you visualize what's happening in the process, and then you'll get to see it again in the video as well. So when you have translation occurring, first of all, the ribosome grabs a hold of the mRNA at the Schindel-Garno. Then picture a tRNA holding the first amino acid, fMet. It's going to come into the ribosome. This will be the only amino acid, the only tRNA that enters the ribosome at the P site instead of the acceptor A site. So the tRNA holding fMet comes in at the P site. Now the second charged tRNA holding a second amino acid will come into the acceptor site. And then I want you to always make the sound pop, kind of like a popping sound, pop, to remind you that the transfer will be the amino acids being held at the P site will transfer to the tRNA in the A site. So that's why I say pa, because the transfer is from the P site to the A site, which you'll see in the video in a minute. Once that occurs, the ribosome then shifts, and all of the tRNA ends up in exactly the right position for what we explained the ribosome sites earlier. So once the ribosome shifts, the tRNA that gave away its amino acids and is now empty and uncharged, that will be at the E site to exit the ribosome. The tRNA that just took the amino acids and is now a growing protein chain, where will that be? That will be in the P site, the protein site, the polypeptide site. And now the A site is open so that the next tRNA can come in, sit at the A site with its single amino acid, and then transfer can once again occur, pop, from the P to the A site. And that will keep happening over and over, which is called elongation. Okay, so these bullet points here show what I was just saying, the P to the A site transfer, and the shift in the ribosome. Then you can't have this go on forever, so termination has to occur. And what termination is when the ribosome reaches a stop codon. And I want you to circle stop codon and remind yourself that there is no tRNA that can read this, which means no amino acid can be brought in to the ribosome once you have a stop codon. Instead, release factor protein, sometimes you'll see it written as RF protein, that binds to the stop codon and allows for the protein chain to be released and the ribosome to be disassembled. Okay, so make sure you repeat this slide once or twice to kind of help you visualize the process of translation. To further help you visualize it, here we have on the next slide a nice little video.
To extend a growing polypeptide chain, the ribosome must select the correct amino acids that are specified by the messenger RNA. An aminoacyl tRNA bound to elongation factor TU, EFTU for short, enters the free A site on the ribosome. If the anticodon of the charged tRNA does not match the codon in the messenger RNA, the tRNA is rejected. The process of trial and error repeats until the correct tRNA is identified. Elongation factor TU hydrolyzes its bound GTP and dissociates. If the tRNA is correctly matched and remains bound for a long enough time, it is committed to be used in protein synthesis. The ribosome catalyzes the formation of the new peptide bond and undergoes a dramatic conformational change. Elongation factor G binds to the ribosome. Hydrolysis of GTP by elongation factor G switches the ribosome back to the state in which it can accept the next incoming tRNA.
So now with these genetic processes, when you think about the processes occurring, there has to be some sort of regulation for them. Keep them in check, right? So prokaryotic gene regulation is very important to the cells. Basically, what do they need at that moment? Do they need the products or do they not need them? And now with gene regulation, it can either be positive or negative. With positive control, think of it as the gene is only expressed if some sort of regulator stimulates it or activates it. Whereas in negative control, the gene will keep being expressed unless it's shut off by a regulator, meaning repressed by a regulator. So positive control, a gene needs to be activated, whereas negative control, a gene needs to be repressed. Now, in this process, keep in mind that prokaryotic genes tend to be organized in operons. And down here, we have a picture of an operon. It basically means multiple genes together under the control of one regulatory region. Now, with any of this kind of regulation, there are two terms you have to be able to distinguish. Some regulators can be cis-acting, whereas others can be trans-acting. What cis-acting means is that the regulator is actually a DNA sequence. So it's a DNA sequence on that chromosome involved in regulation. Whereas trans-acting regulations, when you hear trans, think traveling. Okay, trans or traveling, because these aren't sequences, these are diffusible gene products. These are proteins. So trans-acting regulators are proteins that can swim around and bind to certain genes to regulate them. We'll see in a minute what examples are of these things. Now, like I mentioned a minute ago, when it comes to regulation, you could have things activated or repressed. And so the terms you tend to see is that a regulation pathway can either be inducible or repressible. A lot of times it's the end products that will determine what happens, but it varies pathway to pathway. But anyway, what's important here is to know that gene regulation can be positive or negative, so activating or repressing. That in bacteria, this gene regulation involves what's called operons, so a chain of genes. That the regulators can be cis or trans-acting, cis being DNA, trans being protein. And that it can be inducible or repressible, which you'll see in a minute. Basically, the expression can be activated or repressed. Now, to better understand this, we're going to go through the lac operon. And the lac operon that you see here, it codes for these genes here You don't have to know the details about these particular genes, just know that all three are involved in breaking down and using lactose. Okay? That'll be important to help you figure out what's going on. So with the lac operon, before we go into the details of what everything is doing in this operon, it's easier to figure this stuff out if you first use logic. So here you have an operon that makes enzymes to break down lactose. So think about it. If there's no lactose present at that moment, why would you want to waste resources making enzymes to break down lactose when lactose isn't present to be broken down? Whereas if lactose is present, well then you're going to want the enzymes to break it down and use that lactose, right? So that tells you that lactose is an inducer for this operon, okay? So we're going to go through the key players involved. When you look at this operon, you see a few different titles and names here. The first one is the repressor gene. So whenever you see the letter I in the lac operon, think of inhibitor. The I gene makes the repressor of this operon, of this whole regulation process. P gene stands for promoter, and you already know that a promoter is where RNA polymerase binds to express a gene. You then have the operator, so the O gene stands for the binding site where the repressor will bind, the repressor that was made by the I gene. Okay? You don't have to worry about L. We won't be going into that details at the moment. Okay? So focus on I, P, and O. So when you think of this lac operon, if there is no lactose present, which we see in this figure here, this shows no lactose present. As we said, if there's no lactose present, then you don't need these enzymes. It would be a waste to make enzymes that break down and use the lactose when there's no lactose there. So what happens in this process when no lactose is present, you'll notice that the repressor is bound to the operator. And when you have this big bulky protein bound over here, the RNA polymerase that binds at the promoter, it can't make it through this area to get to the genes to express them. So this operon now is repressed. There's no gene expression. Transcription cannot occur because a repressor molecule is currently blocking the RNA polymerase from making it to the genes. In this final picture here, what you notice now, instead lactose is present. And like we said, if lactose is present, well, yeah, now you're going to want to make the enzymes to break it down and use it. So when lactose is present, what you notice is the lactose will bind to the repressor molecule that was made by the I gene. And so when the lactose is bound to this repressor, it changes the shape of the repressor. We call that an allosteric change. So the lactose binding to the repressor changes the shape of the repressor. So what can't the repressor do now? It can't bind to the operator. And if the repressor can't bind over here, now the RNA polymerase is free to slide its way across all the way through all of the genes and transcribe them, which you see happening here. And ultimately, after transcription and translation, you get the enzymes that can break down and use lactose. Okay, so I recommend kind of pausing and rewinding and going over this slide one more time, just to make sure that you're very comfortable with all of the key players. And if you're having any trouble understanding this, just send me a remind message and I'll try to make it a little clearer. To also help, I made an additional slide, which you'll find on the next slide, that kind of recaps what I just said, some of the key points. So as you can see here, I wrote down some of the key points, so you can pause the video and make sure you write down these notes on your slides. This is just a cute animation which you can find on the Amoeba Sisters website, which has a lot of really good biology GIFs to visualize what's going on. And so this will help you visualize the lac operon and all of the key players. Okay, so here we have camp and cap regulation of the lac operon. Okay, this is going a little bit further in detail beyond the regular idea of the lac operon. This is basically to show you that even though a cell can use lactose and break it down and use it, cells actually prefer glucose, which makes sense because if you think about respiration, what do you always hear? You hear glucose. So if glucose is present, then a cell will prefer to use that and won't want to use lactose. And so what the figures here basically show you how what's called camp and cap, which is glucose dependent, can help regulate the lac operon as well. And even though it's called a form of catabolic repression, you're going to see that it's actually an example of positive control because this is an activating or activator process. So like we said, glucose is preferred. And what was discovered is that polymerases don't actually bind efficiently to the lac operon unless what's called CAP is also present. So CAP stands for catabolite activating protein. And so the way you could think of it is, well, the operon basically thinks that if someone's wearing a cap, they're very attractive, okay, wearing their little baseball cap, very cute, and so it will activate the lac operon. So if cap is present, which we see binding over here, if cap is present, then the polymerase works really well, it binds very efficiently, and you get very good transcription. If cap is not bound to the lac operon right before it, then the polymerase will not be able to bind very well to the promoter, so you won't get a strong transcription because the RNA polymerase won't be that great at binding. So what happens in this process, if glucose is not present, in the absence of glucose, what's called CAMP complexes with cap so that cap can fit perfectly on the promoter. And when cap is present, then the polymerase binds beautifully and you get transcription. If, however, glucose is present, that inhibits the enzyme that makes CAMP. And so that can't complex with cap, and without cap binding to the promoter region, there's nothing there to help the polymerase properly bind. And if the polymerase doesn't bind, then you don't get proper transcription. Okay, so I'm going to recap this on the next slide just like I did with the previous notes. Okay, so here you have a brief recap so you can pause the video and write down the notes in case you missed anything. Okay, so here we have another example of operon control and gene regulation. Here we have the trip operon or the tryptophan operon. Now notice this says tryptophan synthesis. So these enzymes are for making tryptophan. So again, you can use logic and figure, okay, if tryptophan is absent, well then we want to make it, the cell would want to make it, so transcription is going to need to be active. Whereas if tryptophan is present, well then if you already have tryptophan, why would you want to make more? That would be wasteful of resources. So if tryptophan is present, then you're going to want this operon repressed. Okay, you don't want to waste resources making these genes or expressing these genes. Okay, so when you look at these processes, the option B here shows tryptophan absent. Like we said, if there's no tryptophan, the cell will need to make some. So you'll notice that the repressor is not the right shape in this scenario. It won't be able to bind to the operator, so the polymerase will get to bind to the promoter and express the genes. Whereas when tryptophan is present, what it does is it's a repressor. Okay, so the tryptophan will bind to the repressor. It will change its shape so that now it fits the operator. So tryptophan will allow the repressor to bind to the operator, and this blocks polymerase from getting from P through to the genes to express them. So transcription is blocked in this case. Okay, so I'm going to recap this in the next slide just to make sure that you have it in writing and that it's clear. Okay, so here you have the recap. Again, you can just pause the video and take down the notes just to make sure you didn't miss anything. Now, the last thing to mention over here with regard to metabolic regulation is that you can have various forms of regulation beyond, the gene level. So instead, this is at the protein level. Two examples are chemotaxis and quorum sensing. What chemotaxis means is that bacteria can move toward an attractant or away from a repellent. So it's basically a series of proteins will trigger the flagella. So you can see that in this little figure here. These series of proteins will trigger the flagella to move around. Okay, so chemotaxis is just the idea that proteins can either attract or repel a bacteria to move. Quorum sensing, on the other hand, is the idea that basically the bacteria can assess population density. So basically the bacteria will want to make sure that there's a good enough number of cells around it, like other bacteria, before it'll start initiating any kind of expression. Because think of it, if you're a bacteria and you're a single cell with no other bacteria friends nearby, well then if you use these resources to produce a toxin, it's not going to do much for you, right? Because you're a single cell, it's not going to do much to the host, not much done. You just wasted resources. So a lot of bacteria will wait until there's a good amount of other cells present before they start activating these other proteins to be produced or toxins to be produced. So that's quorum sensing. Now, I just want to run through some eukaryotic differences. Even though some of these slides will look scary or complex, we're not going to go into the details of them. You don't have to memorize crazy details with it because this isn't a genetics course. I just want you to be aware of some of the differences that exist for general purposes. So the first difference that you see here is eukaryotic genes are monocistronic, whereas prokaryotes have mRNA that is polycistronic. Basically that idea of the operon that we just saw. You can have multiple genes in a prokaryote squished together that's all under the control of one promoter. Whereas eukaryotes, it's a single gene for a single protein with a single regulatory region. Okay, so this visual just helps you see eukaryotes, single gene, single piece of RNA for single protein, whereas prokaryotes can have multiple genes squished together to make one long mRNA that then gets cut and made into multiple proteins. Some differences in terms of eukaryotic transcription is that for eukaryotes, you see this long list of key players. You have multiple RNA polymerases instead of just one. Okay, you don't need to memorize the 3 exact types. Just know that eukaryotes have multiple RNA polymerases instead of just one. And the eukaryotes do not have a sigma equivalent on their RNA polymerase. I want you to circle, star, highlight that bullet point right here. Eukaryotes do not have sigma. Why is that significant? Well, in prokaryotes, what does sigma do? Sigma sees the promoter. It binds, it allows RNA polymerase to bind to the promoter and start transcription. Eukaryotes do not have that. So instead of sigma, eukaryotes need a series of transcription factors. Okay, so whenever you see transcription factors, these are in eukaryotes to help polymerase recognize and bind or initiate transcription. Okay, so transcription factors serve the purpose that sigma would have, to recognize and bind the DNA to start transcription. This is to show you the various transcription factors that exist. Okay, so there's a whole bunch of them. This big pre-initiation complex needs to form in eukaryotes. Again, it does not need to form in prokaryotes because prokaryotes have what? They have the sigma subunit of the polymerase. So prokaryotes do not need this. Eukaryotes need all of these players to help recognize and bind to the promoter. You don't have to memorize these guys. The only one I want you to know here is this guy here, TF2H. That's a transcription factor in eukaryotes, including your own cells, that will phosphorylate the tail of RNA polymerase to allow the polymerase to move for elongation. So if you visualize this big structure here is a whole bunch over here as well, a whole bunch of transcription factors. What TF2H does is if you picture this big blue blob as RNA polymerase, TF2H will phosphorylate the tail. And if you think about that, I'm going to show you on the next slide why that's important. Because if you think of phosphorylation, phosphate groups have negative charges, right? So that TF2H is putting a whole bunch of negative charges on this piece of the RNA polymerase. So by down here, you can see it. A whole bunch of phosphates is a whole bunch of negative charges on the polymerase. What do you know about DNA and the charge of DNA? Well, DNA, this DNA strand that the polymerase is going to read, is negatively charged. So if you're suddenly adding some negative charges to the polymerase, it'll allow that tail to slightly repel the DNA so that instead of being tightly bound, it'll be able to slide across the DNA and read the gene in order to express it, because you need the polymerase to move across the gene. You don't want it to just stay stuck at the promoter. Okay, so the phosphorylation of transcription factor 2H is very important for the movement of RNA polymerase, which is very important for elongation, for actually making that RNA from the DNA. Okay? So you don't have to memorize all of the other transcription factors I had on the previous slide. Just know the significance of transcription factor 2H is to phosphorylate RNA polymerase so the polymerase can move across DNA and transcribe the genes. Okay? If you're still a little unclear about that, please send me a remind message and I'll clarify it. Now, I want to just make a quick point that all of the regulation that we've seen so far is what we call upstream, meaning the regulation was occurring in front of the gene. Okay? I want you to be aware that in eukaryotes, there are also regulators after the gene. So this is our gene here. These are upstream regulators to the left, and in eukaryotes, you have downstream regulators to the right. What we call these downstream regulators are enhancers. So enhancers are kind of unique because they're far away from the gene that they're regulating. And basically what an enhancer will do is that it will bind, so pictured over here far from a gene, it'll bind transcription factors. And then DNA will loop around, which you see in the picture here. So the transcription factors bound to the enhancer, once the DNA has looped around, these transcription factors are now brought nice and close to the big transcription complex where we have, the activation of the gene. Okay, and remember that in eukaryotes, you can't have transcription without all of these transcription factors, which are not necessary in prokaryotes because, again, what do prokaryotes have? They have the sigma subunit to take care of all of this. I want to point out that gene expression is not the end for eukaryotic RNA synthesis. So another difference between prokaryotes and eukaryotes is that eukaryotes then do a whole lot of processing of their RNA in order to make it mature and usable. Some of the processing that you'll go through or need to know exists is eukaryotes will add a 5' cap, a poly A tail, and splice the RNA. Okay, so when you think of eukaryotes, remember they have to process their RNA, and they do that by adding a 5' cap, a poly A tail to the end, and splicing. So again, with eukaryotes, I want you to remember that they have to process their RNA, that this requires 3 different steps. The first step is that eukaryotes will add what's called a 5' cap, meaning that on the very beginning of the RNA for a eukaryote, they will add this little reverse orientation GTP. Okay? This is important for stability, okay? So this functions as stability because by putting this GTP that you can see here in an upside down orientation, it basically gives the appearance that the 5' end of this RNA is actually a 3' end. The reason why that's important is because in the cell of eukaryotes, there are a lot of 5' exonucleases, meaning that there are a lot of enzymes that will go around and chop up 5' ends of RNA. So by putting a cap, a 5' cap, that makes the 5' end look different, these enzymes can't recognize the RNA and won't chop it up. Okay? It also helps with the movement of this RNA for things like nuclear transport, but don't worry about that for now. Just know the 5' cap gets added on eukaryotic RNA to protect the RNA from nucleases that would chop it up. Okay, so the second step in eukaryotic RNA processing is for the eukaryotic cell to add what's called a poly A tail. So what this means is that on the end of every RNA, the eukaryote will add this very long string of A's. Okay? The reason why it puts this long string of A's is kind of like what we mentioned a minute ago. It's for increased stability of that RNA. It basically helps prevent degradation of the coding region of the RNA. Because think about it, as this RNA is moving around through a eukaryotic cell, like I said, there are going to be a whole lot of enzymes that will try to chop up the RNA, and they usually do it from the ends of the RNA. So by having this long string of A's at the end of the RNA, these enzymes can chop, 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 chop without actually chopping any part of the coding region, any part of the RNA that actually codes for proteins. Okay, so you don't have to know the details of how this is formed, okay, all these bullet points on how it's formed. Just know that eukaryotes have to add a poly A tail, and then it's to protect the coding genes, the coding part of the RNA from getting chopped up. The last processing event to mention is the idea of splicing. And the reason why splicing occurs in eukaryotes but not prokaryotes is that eukaryotes have introns and exons, whereas prokaryotes just have their exons, which are the coding areas. Okay, so in RNA, when you hear exons, those are coding regions. Introns are non-coding. So what splicing basically is that a spliceosome, a special structure, basically needs to cut out any introns and then glue together the exons. Okay, so spliceosomes will cut out any introns and glue together the coding exons. Okay, and to demonstrate what that looks like, we have a little video.
The following animation will describe the process of RNA splicing, an important step in creating the mRNA that is involved in protein synthesis via the process of translation. Key factors in this process include RNA, possessing introns and exons, and the spliceosome. Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
So in a perfect world, the spliceosome would detect a single intron, remove that intron, and move on to the next intron. But sometimes the spliceosomes in reality actually make mistakes, and sometimes they cut more or less than they're supposed to. So you end up with a variety of different proteins depending on how the spliceosome cuts out the introns. Which parts of that gene does it end up removing? Now, I want you to be aware that there are 3 forms of alternative splicing. There's alternative promoter selection, alternative tail site, and alternative splicing via exon cassette selection. Now, in genetics or molecular courses, we go through the exact details of what's occurring. I'm going to give you a few slides now with some, kind of scary figures. You do not have to memorize exactly the steps of it. I just want you to really know that these things exist, okay? So the first one we have here is alternative promoter selection. Like I said, we're not going to go into the details of exactly what's getting cut, what's not. I just want you to know that in eukaryotes, sometimes there are alternate start sites, meaning more than one promoter within a gene, and depending on how the spliceosome cuts things out, will determine which exons you end up in your final product. And each exon is what goes toward coding the amino acids to make protein eventually. So you have one gene, but multiple different RNA possible from it, which means different proteins. Alternative tail site now is, like it suggests, within a single gene, there might be more than one tail signal or end site. So you can end up with different RNA depending on which tail site is selected by the spliceosome when it's cutting. Lastly, you have alternative exon selection. So as you can see, depending on how the spliceosome is moving, you may end up losing some of the exons, and so you get a slightly different protein that'll get produced from this RNA. So like I said, I know it may have seemed quick that I went through these last 3 slides, but again, that's because for microbiology purposes, you don't have to know the details. Just know that one of the differences between eukaryotes and prokaryotes is RNA splicing, and that it can result in alternative promoter, tail site, or exon selection. The last thing I have over here with regard to RNA processing is that it's important for eukaryotic translation, since there's no Schindel-Garneau in eukaryotes and there are introns that you don't find in prokaryotes. Okay, so the RNA processing is important because instead of having a Schindel-Garneau, the way prokaryotes did, eukaryotic mRNA is recognized by the ribosome because of its 5' cap structure that gets grabbed the way the Schindel-Garneau does in prokaryotes. So think of the 5' cap structure as part of eukaryotic initiation of translation. I also put in some bullet points here for you to recognize that sometimes if you're reading any textbooks or online sources, you'll notice that another difference in terms of eukaryotic translation is that the ribosome sizes are different. You do not need to know the size differences, the exact numbers for this class. It's just another point that I want to make, that the difference does exist. The last point that I want to make is the idea of coupled transcription translation. So that's a big difference between eukaryotes and prokaryotes. Prokaryotes can start translating their mRNA right while it's still being transcribed. So this figure that you see here, this is prokaryotes. They'll have their DNA, and then you see the RNA getting made from that DNA. And while the RNA is still getting made, the ribosomes jump onto it and start making protein. Now, why can't coupled transcription translation happen in eukaryotes? Well, think about it. In eukaryotes, are the ribosomes in the same place as the DNA and RNA? No, because eukaryotes have a nucleus. Prokaryotes do not. So in eukaryotes, the DNA and the RNA is in the nucleus. The RNA then has to leave the nucleus and find the ribosomes out in the cytoplasm and the rough endoplasmic reticulum. Okay, so when I ask on this slide, why is this not possible in eukaryotes? The answer is coupled transcription translation is not possible in eukaryotes because we have a nucleus. So our ribosomes that would make the proteins or translate are not in the same place as the DNA and the RNA. Okay? If you're unsure of what this, what I was just saying about this, again, feel free to send me a remind message. So, as always, any questions, any concerns, anything that you think I went too fast through, or that you just want some extra practice or information on, always feel free to send me a remind message. Now, what I want you to do is head over to the discussion board, and each student must post the 2 questions that you would have written on the board. Okay, so 2 questions from today's lecture slides. Okay, I hope you enjoyed the online lecture and have a great day.